Okay, well, good afternoon, everyone. I think as we've got a very full agenda for an hour, we should get going pretty much on time. As others join, they can, I'm sure, pick up the thread. So welcome to today's webinar, where we're celebrating our two winners of the inaugural MPCCC Award for Outstanding Cancer Research in Victoria. My name is Melissa Southey and I'm the chair of the executive for the Monash Partners Comprehensive Cancer Consortium. I'd like to begin by acknowledging the people of the Kulin Nations, who are the traditional owners of the land on which we are gathered today at the MPCCC and across Victoria. I respectfully acknowledge their elders, past, present and emerging, and I pay my respects to any members of the Aboriginal and Torres Strait Islander community gathered among us this afternoon. Our search for Victoria's most outstanding mid-career cancer researcher resulted in two exceptional researchers from the Monash Biomedical Discovery Institute being jointly awarded the winning prize. Dr. Faridi is a research fellow from the Department of Biochemistry at the Monash BDI, and Associate Professor Taylor is the deputy head of the Monash BDI's Cancer Research Program. Doctors Faridi and Taylor have each had their research published in prestigious journals over the past 18 months, and the MPCCC is delighted to recognise their achievements and support the development of their careers with a $5,000 prize. Our independent review panel, who selected Drs. Faridi and Taylor as winners of the award, also nominated three honourable mentions. They are Dr. Sarah Best, who's a Senior Research Fellow from the Walter and Eliza Hall Institute of Medical Research. Dr. Jessica Hollian, who's the Vice Chancellor's Fellow from RMIT University. And Dr. Rebecca Engel, who's a Senior Research Fellow from the Cabrini Monash Department of Surgery and a Postdoctoral Fellow at the Monash Biomedicine Discovery Institute. I would like to personally congratulate our winners and those receiving honourable mentions for their contribution to Victoria's cancer sector and the importance of their research and ongoing advancing not just Victoria's cancer research, but cancer research internationally. The MPCCC were delighted to receive such impressive nominees to our inaugural award and believe this demonstrates a bright future for cancer research in Victoria. Just before we begin, I'd like to go over a few housekeeping issues for today's webinar. Could all attendees please ensure that you have your video turned off and your microphone muted before we begin? I will be taking questions at the end of the presentations. To ask a question, please click your participants button at the bottom of your screen and raise your hand. Please remember to unmute and introduce yourself when you're asking a question and to lower your hand when you're finished asking your question. Over the next hour, we will hear from both Drs. Faridi and Taylor about their research, and we will also hear from their mentors who nominated them for the award. Professor Roger Daly and Professor Tony Purcell, and to hear insights into the contributions these two individuals have made to research in recent years. At the end of their presentations, we will hear from our winners about how they have reached this stage of their career and what impact they believe this award will have on their future research. So without any further delay, let me hand over to you, Tony, Director of the Department of Biochemistry and Molecular Biology at Monash University, and allow you to introduce Dr. Faridi.
Thanks, Melissa. Thanks for the promotion. Didn't realise I was the director. Sorry, Roger. Look, it's a pleasure to introduce Pouya today. Pouya joined my lab in September 2016 at a point when our field was sort of being turned upon its head in some respects. So we work on antigen presentation, and we were sort of, I guess, confronted with some big changes in how we think about the sorts of peptides that are recognised by T cells, and in particular, two papers published in Science late that year, both pointing towards quite a prominent role of spliced peptides or hybrid peptides in various forms of immunity. So Pouya really sort of grasped the opportunity to sort of really try and understand these peptides, where they come from, and what their role may be, particularly in cancer. I'm sure that's what he's going to talk to you today about. I nominated Pouya for his recent paper in cancer immunology research, which followed up from a very nice Science Immunology paper published in 2018. I think one of the really notable things about Pouya is his sort of endless enthusiasm. Nothing really phases him, and he's really not afraid to ask questions. In fact, he asks me a very long list of questions virtually every day I see him. And that's really led to a really exciting collaboration with Professors Jerry Lynette and Beatrix Carino from University of Pennsylvania and the Parker Center for Immunotherapy, where some of our initial findings are actually going sort of first into man, clinical trials using dendritic cell-based vaccines and these spliced peptides to treat melanoma patients. So without further ado, Pouya, looking forward to your talk.
Oh, thanks, Sonny, for a very nice introduction. And thanks, MPCCC, for giving me this opportunity to talk about my research today. And obviously, thanks for the prize, which will be very useful for my future, which we'll discuss it later. So today I'm going to talk about spliced peptides and how we can use them in cancer immunotherapy. So cancer immunotherapy, I'm sure most of you heard about cancer immunotherapy. It's a new method that people use in 21st century, and there are lots of patients that relieve from cancer because of having access to this technology. So in principle, cancer immunotherapy means harnessing the immune system to fight cancer and ultimately increase patient survival rate. So there are lots of reports about increasing survival rate in different kinds of cancers, such as melanoma, breast cancer, colon cancer. And to acknowledge the impact of this approach, in 2018, two scientists from two sides of the world, from James Allison from the United States and Tasuka Honjo from Japan, received Nobel Prize for their research and discoveries in this field. There are different approaches in cancer immunotherapy, but the most prominent approach is using T cells, so which is a kind of immune cell in our body. So the way that T cell can be used for cancer immunotherapy is that we have an antigen processing and presentation pathway which dynamically presents what's going on in the cell to the cell surface. So the way that it does this job is that proteasome digests intracellular proteins to peptides, and these peptides bind to a molecule called HLA, and this complex comes to the cell surface. And T cells constantly monitoring these complex. So in case we have a peptide which particularly present in tumors, such as peptides that carry a mutation carry a mutation which is specific to cancer, or peptides derived from cancer germlines or cancer-specific antigen, or peptides that present from endogenous retrovirus that only express in cancer situation. So if T cell find them, they recognize it and lyse the cell. So this is the fundamental of immune recognition by T cells and how we can use this for cancer immunotherapy. So one approach which is now very well known and different people use this approach is that we can take dendritic cells from patient and then expand them, mature them, and then by selecting some of the peptides which are cancer-specific peptides, we pulse these dendritic cells and then inject them back to patient. So as you acknowledge, selecting the right peptides for pulsing dendritic cells is very important. It's somehow the key factor in this approach, and we usually can use maximum 10 to 12 different peptides for this strategy. So it's very important to find the best peptide, most dominant peptide, and most immunogenic peptides for this approach. So where these peptides come from? So for long time people believe that proteasome, which is the main peptide generation machinery in our antigen processing and presentation pathway, can cleave intracellular proteins to peptides, which are linear peptides. It means that they have template in their original protein. But as Tony said, some papers published in early 21st century showed that there are some reports that proteasome not only can cleave or cut proteins, but also can ligate small peptides together and make a new peptide, new sequence, which doesn't have any origin in our proteome or in genome. So these two segments could come from same protein, which in this case we call it cis-spliced peptide, or could come from two different proteins, which we call these trans-spliced peptides. And this was a big technical challenge. The reason it was challenged that the method of choice for identification of HLA bound peptides is mass spectrometry, and most of workflows in mass spectrometry are somehow based on using a proteome template to identification of peptides. But we don't have any template for cis or trans-spliced peptides, so it was a big challenge to identify these peptides. So what we did, we developed a workflow, mass spectrometry, bioinformatics workflow to identify these peptides. So we use a technique called de novo sequencing, which we could sequence peptide without having any template and find spliced peptides candidates and make a database for spliced peptides in addition to linear peptides.
Understanding how these drugs
and then use available proteomics workflows.
That's fantastic. And how many people do you have in
and have this workflow for identification of linear, cis, and trans-spliced peptides. So we call this approach hybrid finder, and we implemented this approach in the general workflow for HLA peptide identification, or what we call it immunopeptidomics. So if we have a cell line or a tissue which present HLA peptide complexes, then we use immunoprecipitation to pull down this complex, dissociate peptides, use HPLC for fractionation, and then run it on mass spec. And now, by having our workflow, we can see and we can identify linear, cis, and trans-spliced peptides. So we developed this technology, and the first question that we wanted to answer was, how prevalent are these peptides? So we have used 17 monoallelic dataset cell lines that express different HLA allomorph, as you see here, to see what's the prevalence of cis and trans-spliced peptides in each of these cell lines. And as you can see, sometimes up to 40% of the peptides that present on the cell surface are spliced peptides, which means it's a very, very big fraction. So we use several different approaches to validate these peptides. I just showed you two of these approaches. So first, we synthesize some of these peptides and compare the mass spectra from eluted peptides and synthetic peptides, and we showed they are almost identical. And on the biological side, we, in collaboration with Professor Jamie Lastron and Dr. Julian Vivian, we solved the structure of these peptides with their corresponding HLA and showed that these peptides can bind and can make this structure. So having all this together, we proposed a new model for antigen presentation and peptides that present on the cell surface. And we showed that in addition to linear peptides that most of people are looking for that, we have also cis-spliced peptides and trans-spliced peptides. So up to here, we showed that these peptides are real and they have a high proportion of immunopeptidome. But the question is, what's their role in immune system? So we started a collaboration with Dr. Andreas Bechernet from Olivia Newton-John Cancer Research Center. So they have a great cell line melanoma cell line panel, patient-derived cell lines. And we started our work on melanoma. So we choose one of the melanoma cell line here, LML44, in the presence and absence of interferon gamma, somehow to mimic the tumor microenvironment, and then use the same workflow that we had for identification of linear and spliced peptides. But the main focus for this project was finding cis-spliced peptides that derive from cancer-specific antigens. So the first observation was, regardless that if we have interferon or we don't have interferon, something around 6 to 8% of peptides that present on melanoma cell lines were spliced peptides, cis-spliced peptides. But as I said, our main focus was on cancer antigen. So here you see this graph shows that every single antigen that is in x-axis is one of the known cancer antigens. And in y-axis, you can see the number of peptides that we found from each of these antigens. As you see, in most cases, each antigen can be represented by both linear and spliced peptides, but in some cases, antigen just present in spliced form to the cell surface. And if we don't look at spliced peptides, we can't see these antigens all in our discoveries. So this showed that almost 30% of peptides that we found from cancer antigens were spliced peptides. So far we know that these are spliced peptides from cancer antigen, and for next step, we want to see if immune system can recognize it. So we selected 30 spliced peptides derived from cancer antigen and pooled them to 3 different groups. So each pooled 10 peptides, and we got melanoma patients PBMCs, which contain their T cells, and also some healthy donors PBMC to see if their immune cells, in particular T cells, can recognize these peptides. So I mean, in a very simplistic way, we pulse PBMCs with these peptides, and again pulse them after 10 days, and then we read if these T cells can express inflammatory cytokines like TNF-alpha or interferon gamma. So what you see here in A is that different studies that we use as a control, you see we have negative controls and positive controls. And when we have the expression of TNF-alpha, it means that the immune system can recognize the peptides. In the first step, we looked at different pools, and we found for all 3 pools, immune system, T cells can recognize at least one of the peptides in this pool, and then we deconvoluted each pool to see which of that peptides are exactly the immunogenic peptide. As you see here, for example, for pool 1, after first pulse, in the second pulse, after 10 days, we used individual peptides, and for example, peptide 1 was not immunogenic, but peptide 1832 was very immunogenic. So we found these peptides, some spliced peptides, which are immunogenic. Actually, we found 6 cancer-specific immunogenic spliced peptides. So the other question that we wanted to answer was, if spliced peptides is something very personalized, or we can see same spliced peptides across multiple tumors and multiple patients. So what we did, so we had this data set from LML44, then we used another cell line from same patient, but this cell line derived from a tumor at a different stage of cancer, 2 different stages of cancer, and we analyzed that data set, and we see a good overlap between them. And then we choose another cell line, so this time from a female donor, but completely different patient, and we also analyzed that one. And when we compare the peptides, spliced peptides derived from cancer antigens in 3 individual tumors, we found more than 100 shared cancer-specific spliced peptides between these 3. And interestingly, some of the spliced peptides that we found immunogenic were set in these 100 peptides. So it means we found some peptides that are dominantly present on different tumors and also immunogenic. So having access to this data set, we thought about what's the next step. So can we do something with this? Can we treat some patients? So we started a collaboration with Professor Gerald Linnett and Beatrice Carnot at Parker Institute, University of Pennsylvania. So they are leaders, both leaders in dendritic cell vaccination, as you see their science paper published in 2015, and they showed that the approach that they have, they can treat patients when they use neoantigens in dendritic cell vaccination. So their approach was based for finding cancer mutation and make peptide which carry the mutation. But as I said, one of the problems for looking at mutation is that it's very personalized. And if you make a vaccine for one patient, you cannot use it for another patient. So we started this collaboration, and this is the first human clinical trial on using spliced peptides for cancer immunotherapy. And you can see this already started. 2 patients received the vaccine, and hopefully by end of this year or early next year, we will see the results, hopefully very good results. So, I mean, we are working on other cancers as well. We are working on some pediatric brain tumors like DIPG, breast cancer, mesothelioma, which we will have a clinical trial on that as well. Sarcoma, neuroblastoma, and colon cancer. As you see, this is like a workflow that we can apply on different kinds of cancers. As a summary, we have developed a workflow for identification of spliced peptides, and by using that workflow, we showed that spliced peptides have a high prevalence in immunopeptidome. We showed that spliced peptides are not very personalized and most like a general targets for cancer vaccination and can present by different patients. And we showed the immune system can recognize them, which means they are very good targets, or at least possible targets for cancer immunotherapy. So I would like to thank, first of all, Tony for his support on everything. And Personal Lab team, also Jamie and Julian from Monash University, Andreas, which most of the work that we have on melanoma, we work together. Also other collaborators, especially, and also Jerry and Beatrice, it was a great opportunity that we can start with this clinical trial together, and also NHMRC, JDRF, and Mark Hughes Foundation for their support. Thank you very much.
Thank you so much, Pouya. It's clear why your nomination was so highly ranked by the assessors. That's inspirational to hear. We won't stop and discuss this at this point. I think we'll move on, and I'll introduce Roger Daly, please, to introduce Dr. Renee Taylor and to tell us why he nominated Renee for this award. So to you, Roger.
Thanks, Melissa. Yeah, it was obviously an absolute pleasure. It's an absolute pleasure to nominate Renee for this award. And indeed, there was quite a few studies and papers which Renee's been involved with, which could have led to this nomination. But the particular paper I chose was one which Renee and her colleagues published in Science Translational Medicine. And this was identifying a kind of a novel vulnerability, if you like, in prostate cancer cells, where they have a dependency on fatty acids for their metabolism. And I think this was particularly interesting for a number of reasons. I mean, one reason was that altered cancer metabolism has really come to the fore as one of the more recently emerging hallmarks of cancer. And so it was kind of interesting to see this concept applied to prostate cancer. The second aspect of it, which I thought was very important, is that this particular vulnerability can lead to a targeted therapeutic strategy, because they actually demonstrate in this paper that using an antibody called CD36 against the fatty acid transporter represents a potential therapeutic strategy. And then kind of a third highlight of this particular study, although it's one which you could apply to all of the work really which Renea and her collaborator Gail Risbiter are doing, is that exploits the generation of incredibly powerful patient-derived models of prostate cancer, both patient-derived xenograft models, and that group have established an incredible bank of these, which provide very powerful models for disease subtypes, and also patient-derived organoid models. So yeah, there was a number of reasons for me nominating this particular paper, but as I said, there's a number of other studies from Renea and colleagues that could have been nominated as well. And I just think generally also Renea has really emerged as such a leader and role model for female scientists in not only in Victoria, but also nationally and internationally. So I think she's a very fitting recipient for this award, and I was absolutely delighted to nominate her.
Thanks very much, Roger. I think at that point, Renee, we can hand the share over to you. Thank you.
Wow. Thank you, Roger. That was really a very kind introduction, and I think you've just given a perfect introduction to my talk today. So it was like it was scripted. But I'd like to thank you for not just that kind introduction, but also for nominating me. The MPCCC has been a great supporter of our research discipline at Monash BDI, and for me personally has allowed me to interact with the broader clinical and cancer care network at Monash, and I'm absolutely honoured to be recognised in this way by the MPCCC. As Roger said, the award is based on a publication from my work in obesity and cancer metabolism, and that's what I'll focus on in my talk today. But before I do, I just wanted to take a moment to share a bit of my career history with you and identify some of the really pivotal moments that have led to me being here today, and reflect on how the Monash community has really shaped my career. So I first joined Monash in 1994, showing my age, after finishing VCA studies in Gippsland, and I came here to do a Bachelor of Science and Honours, and then went on to do my PhD at the Institute of Reproduction and Development. This was the first time I really started studying the prostate gland. Following on from my PhD, I went to study human embryonic stem cells in the National Stem Cell Centre, and was learning to apply my skills to prostate cancer. And this was probably one of the most pivotal moments in my career, where following that appointment, I was awarded a 4-year fellowship from the Prostate Cancer Foundation of Australia. That came at a time when I had several career disruptions. I was starting to have children, but the PCFA picked me up and supported me as a young investigator, and made sure I stayed in prostate cancer for the rest of my research career. After that, I went on and took an academic teaching and research position in the Department of Physiology. And ironically, I started teaching the same unit that I had taken back in 1995. So life does sometimes go in full circle. And then, look, the second pivotal moment was more recently when I was awarded the Mid-Career Fellowship from the Victorian Cancer Agency. And that was, again, an opportunity for me to continue my research in prostate cancer. It gave me teaching relief, and it really allowed my research career to flourish. And at the end of that fellowship this year, I'm really proud to say that I'm been promoted to Associate Professor, and I'm leading a very successful research program in the BDI. So through all of that, there's been one person who supported me and now co-leads the prostate cancer program with me, and that's Professor Gail Risbridger. Her contribution to my career has been immeasurable, and today I would particularly like to thank her. So the cancer of interest in my lab is prostate cancer, and this is the most commonly diagnosed cancer in males, with over 18,000 new cancer diagnoses every year. It's also the second leading cause of death in men, second only to lung cancer. Now, the thing about prostate cancer is it's a relatively slow growing cancer, with patients experiencing an average lifespan of about 10 to 15 years post-diagnosis. Yet despite this, we still lose about 3,000 men to prostate cancer every year in Australia. Prostate cancer has a very typical disease course, transitioning from an organ-confined or localised disease state through to an advanced or metastatic stage known as castrate-resistant prostate cancer. And this is the lethal stage of the disease. So the focus of our research program is to try and identify risk factors that promote the transition from a slow-growing and indolent localised tumour to these more aggressive metastatic and therapy-resistant tumours. And our philosophy is that understanding the biology behind these risk factors may be pivotal in helping us identify possible preventative strategies, or at least improve treatment options for men who have these aggressive tumours. So my research program is centred around three main pillars. The first is family history, and we've published a large body of work examining the impact of BRCA2 mutations in men with prostate cancer, and our ongoing studies in this area are trying to identify other genetic risk factors for prostate cancer. The second is in pathology, and our lab has identified several hallmarks down the microscope that can possibly refine prostate cancer diagnostics and improve treatment plans for those men with these aggressive features in their cancers. The third pillar and the focus of my presentation today is our work in obesity and cancer metabolism. So we're all aware that obesity is a pandemic impacting about 2 billion people globally. And obesity and metabolic disease are considered umbrella terms that describe excess adiposity, insulin resistance, and dyslipidemia. And there's an ongoing need in the field to understand how obesity and its comorbidities contribute to increased mortality in patients. And in my program, we've been focusing on understanding the links between obesity and prostate cancer. I'd really like to acknowledge generously at the outset that this program of work has been co-led by Professor Matthew Watt from the University of Melbourne, and Matt brings expertise in obesity and lipid metabolism to this project. So the extended survival time of prostate cancer patients mean that there are hundreds of thousands of Australian men living with prostate cancer, and they're living in the setting of an increased prevalence of diabetes and obesity. So prostate cancer is diagnosed most commonly in men over the age of 55 years. And you can see the prevalence here shown on the slide about type 2 diabetes significantly increases in men of this age group, such that 20% of males have diabetes by the age of 75 or older. And this is the typical age of patients dealing with prostate cancer. Interestingly, the epidemiological data show that men are no more likely to develop prostate cancer if they are obese or have type 2 diabetes. However, there is overwhelming and consistent data from multiple prospective cohorts showing that men who are obese, when they are diagnosed with prostate cancer, they have a more aggressive prostate cancer subtype, resulting in poor prostate cancer specific survival compared to lean patients. Thus, we consider obesity and metabolic disease to be a driver of prostate cancer progression. And we really think that understanding the biological mechanisms that underpin this association are required in order to improve patient outcomes. So there are numerous physiological changes that happen in a person when they develop obesity, and many of these have been postulated to contribute to cancer pathogenesis. Several years ago, Matt and I summarised some of these factors in a review article, identifying a number of systemic changes in obesity that can impact on cancer pathogenesis, such as insulin resistance, low-grade inflammation, and altered steroid production, which is particularly relevant in prostate cancer. There are also many local changes, and these particularly happen in the adipose tissue itself. And one of these things is dysregulated lipid metabolism. And understanding how these changes in the adipose tissue impact prostate cancer progression has been a focus of our studies. So in order to fulfill the many physiological requirements to support tumour growth and metastasis, cancer cells have an increased need for energy provision. And there are several substrates shown in this schematic that are available to cancer cells to fulfill some of these growth requirements. We know that glucose is a major substrate for cancers through the well-known and well-described Warburg effect, but this is not the case in all cancers. And particularly in prostate cancer, we think there might be other more important substrates, and we'll be focusing on the role of fatty acids. So one of the reasons that we postulated that this might be a preferred substrate for prostate cancer is the close proximity of the periprostatic adipose tissue bed that sits anteriorly on top of the human prostate gland, and is potentially important because this is an adipose tissue bed that would store and release fatty acids. So clinical and anatomical studies have shown that as the thickness of the periprostatic adipose tissue bed increases with increasing adiposity and obesity, so too does the likelihood that men will be diagnosed with prostate cancer. And more specifically in the green bars is that this cancer will be of an aggressive phenotype that has a likely to have poor outcome. Secondly, we know that the adipocytes in that tissue bed sit in quite close proximity to the cancer itself. And in aggressive cases, the cancer cells can actually be seen to migrate out into that periprostatic adipose tissue where direct cell-cell contact can occur. And so for all of these reasons, the periprostatic adipose tissue is certainly a prominent and likely source of fatty acids to prostate cancer, particularly in obese men. So in order to test this hypothesis that fatty acids are a metabolic substrate in human prostate cancer, we obtained patient-matched samples from patients undergoing radical prostatectomy. Our lab, through collaboration with clinicians and pathologists, has access to a couple of hundred patient specimens per year, and this forms part of a national tissue bank for prostate cancer. So for this experiment with these fresh tissues, we perform metabolic analyses using radiolabeled glucose and fatty acid traces. That's an expertise of Matt and his lab. So I don't have time to show you all of the data today, but just focusing on the results from the fatty acid tracer, where we saw a number of highly novel findings. So firstly, we saw a significant increase in the uptake of fatty acids into prostate cancer tissues compared to patient-matched benign tissues. And this increase was observed in all of the patients we tested in this cohort. Interestingly, there was no concurrent increase in fatty acid oxidation or utilization for immediate energy provision. And really that led to a mismatch between the fatty acids that were uptaken and those that were used for energy, suggesting there might be a greater conversion of fatty acids in the cancer cell to other types of lipids. And indeed, a detailed analysis of lipids by chromatography showed that there was specificity in the partitioning of these incoming fatty acids. And specifically, these fatty acids were incorporated into lipids that involved in membrane protection, such as ceramides and phospholipids, and these were significantly increased in cancer. So we have really shown that exogenous fatty acids are a source of energy provision for prostate cancer, and it's not that they're immediately utilized, but actually contribute to the production of complex lipids that can be used for other physiological functions. So in order to study the mechanism of fatty acid uptake, we turned to the major fatty acid transporter, CD36, and examining the publicly available databases, it was very evident that a high level of CD36 expression was associated with reduced survival in prostate cancer patients. And further examination of other genomic data sets show that CD36 is most frequently amplified or gained in human prostate cancer tissues, and we very rarely saw loss or deletion of this gene. So to validate our findings, we generated a novel mouse model, which was on the background of the Proteasome P10 knockout mouse that specifically develops cancer in the prostate gland. And we crossed these to a CD36 flox mouse to generate a double knockout mouse in the setting of a cancer background that was deficient in CD36. We confirmed the loss of protein in these prostate tissues, and functionally, most importantly, we showed that knocking out this single transporter was enough to prevent the uptake of fatty acids back to wild type levels. So using this mouse, we tested the hypothesis that CD36 ablation could reduce prostate cancer progression. And indeed, this was the case. So we saw a marked reduction in the size of the mouse prostate gland at 6 months in green here in the CD36 knockout mouse, compared to the P10s, which develop frank malignancy and the prostate glands are quite large. And probably more importantly, looking down the microscope, we saw that there was a halving of the incidence of cancer observed in those tissues, denoted here as carcinoma in situ and shown in the black bars, where less than 25% of the animals had any evidence of invasive carcinoma. So when coupled with a reduced tissue size, this CD36 knockout mouse really does have a substantial reduction in tumour burden. So as Roger mentioned, we then went on to utilise some of our preclinical models that we develop in our lab, and to assess the effect of the therapeutic potential of blocking CD36, we turned to our patient-derived xenografts of localised prostate cancer. And we specifically use this CD36 monoclonal antibody. So these patient-derived xenograft studies we used two particular PDXs, one that was very highly reliant on fatty acids, and one that didn't really show a high reliance of fatty acids. And as you can see from the data, the CD36 monoclonal antibody reduced the tumour growth in the line that was particularly reliant on fatty acid uptake. So this was the first time that we had preclinical evidence that blocking CD36 can potentially be exploited for therapeutic gain in human prostate cancer. So as many of you would know, the major clinical challenge in our field is to prevent men with a moderate or a high risk localized disease developing metastatic or advanced prostate cancer. And our preclinical studies in mice and in PDXs have shown that CD36 deletion, in fact, at an early stage of disease can delay the progression to advanced prostate cancer. And as Roger mentioned, that is the reason why this paper made it into Science Translational Medicine. It's because of the therapeutic potential of blocking this particular fatty acid transporter. And as the field advances and better drugs are available to target CD36, we'll definitely be looking to see if we can translate this into the clinic, into early clinical trials. So beyond these initial findings, we've been exploring whether this reliance on fatty acids is similar or different in the setting of chemotherapy or other systemic treatments for advanced or metastatic disease. And towards this goal, we've recently been performing metabolic profiling on a cohort of our PDXs. As Roger described, we have a large cohort now of more than 50 PDXs in our lab that represent both localized and metastatic prostate cancer. So what you can see from this heat map is that there's significant metabolic heterogeneity in individual PDXs. Each PDX is represented in the columns here, and each of the substrates being utilized is shown in the rows. So the first observation that we failed to observe any clustering between localized or metastatic samples, indicating that there's no clear switch in metabolic substrate utilization as metastasis occurs. But what you can also see that there are subtypes of tumours that have a high reliance on fatty acids, as we previously described. There are others that do clearly utilise glucose and others that prefer to use glutamine. And potentially most interesting are the subset of tumours that seem to not rely on any of these major substrates. And these are generally the more rapidly growing PDX lines. So our future work is really aimed at trying to determine what other possible substrates could be active in this particular setting. And I think the future is quite exciting in this space. So in summary, our data published in the paper showed that fatty acids are a major substrate fueling prostate cancer tumorigenesis, and obviously offer a viable therapeutic target if we can get the right drugs into the clinic. Subsequent studies, we've identified marked metabolic heterogeneity between prostate cancer tumours, one that probably had not been appreciated before. And really our job now is to try and understand which is the substrate that should be targeted at which time in stage of disease progression and in which patient. And therein lies our challenge for the next phase of our research. So just before I finish up, I do want to take a minute just to share one of the more pleasurable parts of my job, and that's engaging with the prostate cancer community. It's really an honour to be associated with some of these fabulous organisations listed on this slide, especially the E.J. Whitten Foundation, who directly support our research program at BDI, as well as these other funding partners who generously allow us to interact with their patient networks, disseminating our research findings with the community, raising awareness of prostate cancer. And for me, this has a major impact beyond the laboratory, and it's a really very enjoyable part of my job. So finally, I'll just finish by thanking again my entire research team at Monash and at Peter Mac, especially Gail Risbridger, Matthew Watt, and also Mark Frydenberg, who's been my clinical collaborator since the beginning of my studies. Thanks also to these funding bodies and the patients and families who generously donate to our prostate cancer research program. And finally, just thanks again to the MPCCC for generously bestowing this award on me. Thank you.
Thanks very much, Renee. A great illustration. I guess just some of the breadth and depth of research that's going on, not just under your own steam, but within a hugely diverse group of collaborators and colleagues. So thank you very much for a small insight into your work. We have some time now for a short discussion, and Renee, you gave us some idea about what were pivotal steps in your career, but Pouya, I wonder if I could throw this back to you and ask you you know, are there really key moments where it really assisted you to progress in cancer? We know, well, not just in cancer, but perhaps in research more broadly, we know that this is a pathway fraught with dangers, and we lose so many of our colleagues who don't progress in the pathway that we perhaps think they should. It's interesting to hear what's pivotal for people so we can try and reinvent others who are coming behind.
Sure. So the way that I get here at Monash is a little different with most of people. So I started pharmacy and PhD in Iran, and then I went to Zurich and Switzerland. I was there for almost a year. I went back to Iran, and 4 years ago I joined Toyn's lab. Before joining Toyn's lab, I was not working on cancer. So actually I joined Toyn's lab for starting work on one of his projects on cancer, antigen discovery in cancer. So in this last 4 years, it was two specific times that I was super, super happy. So when I wanted to start, working on spliced peptides, as I said, there was no workflow for that. And I designed and tried maybe 40 different workflows, and they didn't work. And finally, one of them worked, and we found a peptide that we could confirm it. And I mean, you can't believe how happy I was when I found, okay, after 40 different workflows, one is working. So this was the first. So I was very happy. And then when I got an email from Andreas that some patients, immune cells can recognize one of these peptides, which means, okay, so we really have something that we can use for patient treatment, which is, I mean, it's like science always like to have a real impact on society and patients. And that was the second moment. And then I had lots of dreams that I will get a phone call from Jerry that, okay, patients don't have cancer anymore. And I'm waiting for them. Yeah, that was, I mean, I think that was the highlights of my career for the last 4 years.
Yeah, I suppose you're still in the position where you still wait for that news. It's going to be an ongoing. You're waiting for news.
Yeah.
Huh. And Pouya, is the prize going to have some assistance in this process of moving forward from here? Is there some practical or left field idea that you have that the prize money may actually be helpful?
So definitely, I mean, so I think the concept of a spliced peptide is still quite new, and I need to introduce this to other scientists. Definitely, I will use this to present my research in different conferences. And the good thing for COVID-19 somehow is that we can stay in Melbourne and present our studies all around the world, so we can participate in different conferences, like online conferences. So I think my plan is use this money, not all of them, but some of them, to present my work in 2021 at different conferences and introduce this concept and idea to other scientists.
Yes, when we started to think about the award, I suppose many of the executives thought that it might be used for travel. It enables someone to attend a meeting that wasn't going to be possible. We didn't at the time envisage that we would be in this situation at this time. And, you know, virtual meetings are great. And actually one of the things I'm finding about virtual meetings is that a lot of younger researchers who wouldn't have ever been able to attend meetings are able to attend virtually. But I think what that misses is the opportunity to establish collaborations, you know, the sorts of conversations that you have at coffee breaks at meetings.
Yeah.
And I think, Renee, both you and Pouya in your talks presented that, I think, tell me if I'm wrong, that collaboration has been an important part of both of your progress. And I worry now that we're going to have this period of time where some of our younger colleagues aren't going to have that capacity to develop collaborations like you two have, and perhaps even sustain them. Renee, I'm sort of dropping this on you at this point, but have you thought about this? Do you think that there's ways that we can negotiate the next 12 months at least ahead?
Yeah, I mean, I think, Melissa, as you said, because travel is not possible, it's made you think about what to do with the money and how it can help. And I think one of the things I've recognized is that it's my job now to give back to my young people who I'm training and afford them the opportunities that I had in the early stages to be exposed to different networks and opportunities. So hopefully there'll be a chance for me to do that for my young people, because I absolutely recognize that, you know, it's these conferences, I've just spoken at one this morning, you know, that you write the question in the chat, there's no room for interaction. You certainly don't get to meet people afterwards. So hopefully sometime next year that can be changed. But again, just giving them opportunities to train and think about, you know, their abilities and work out where their strengths are and help build their portfolio is something I'd like to focus on in the next 12 months while we do have that quiet time together.
Yes, I think many of us have been thinking about that, and I'm sure the executive of the MPCCC and the VCCC actually would be interested in knowing if you have any great ideas. We don't want to be focused just on the MPCCC domain, but if there are across Victoria activities we can do in that space, that would be really interesting to hear from you guys and from anyone who has suggestions about how to make those things cohesive in a time where they're not naturally happening. Renee, you talked about your family very briefly in the beginning. I wonder if you'd like to make any comment about progression in science as a female researcher. I don't know if I'm going in territory here that's dangerous, but it's territory that I stand on as well. So tell me if I'm inappropriate. But I don't know whether you think about your younger people when you.
Yeah, I think that's one of the things I was reflecting on, and you know, that reflection was as much for me as to share with the audience today. And you know, there was a couple of moments there where I could have been lost to prostate cancer research, you know, having young babies and not the, you know, the continuity of publishing, and, you know, there was some pretty lean years there. But, you know, I had a supportive network around me at that time. And these grants that you get as young investigators are the one thing that see you through. And the good thing about those two grants is that they were both 4 years long. So it was long enough to get in and be productive over it, you know, take the pressure off some of these short-term grants. So look, I have 3 children. I've been balancing work life for a long time, but it's doable, and you know, we have to make the choice to make it work. I've just been at home for the last 6 months more than I ever have in my career. I'm quite happy to be back in the office. But it's not just young females too. There's lots of families that have things to juggle outside of work. And I think, you know, this is a type of career where there is a lot of pressure. We need help remembering what's important at times. But certainly I'm a big advocate for having a support network around you. So I had, you know, even when I was out of action for a while, there was people that could pick up the pieces so that it was seamless me moving back into the lab. So I'm really very thankful for that. The other thing is, you know, generations of scientists. So, you know, I've highlighted Gail and Mark today, and they started working together 20 years ago. But I can see David Pook and Mitch Lawrence in the audience, and they're now my peers. And, you know, we're almost like children of our senior colleagues. And now we have our own younger peer support network. And I think that you just can't understate how important that is to have colleagues at the same age and stage to interact with and go through your careers together. So I'm very thankful for people like Mitch and David.
Well, I think life is life, and working in that sort of environment helps everyone, I think, including research progress. I don't know that that's a particularly gender specific phenomenon, but we're still faced with this issue of it obviously being much more challenging for women in our field. So I think that's a really nice response, and sorry if it was caught off guard. Anyway, I've been uttering too much. I wonder if other people might like to weigh in. We do have a few more minutes for discussion. So maybe I could ask the future question then. You've talked about what were the highlights. I mean, what would be the fantasy in terms of what the next highlight would be for both of you?
Well, I want to say seeing my work be translated into patient care, but I think Pouya's already on the way to do that. You know, we spend our whole careers looking at discovery science and hoping we'll find something exciting. And that's why I'm working closely with Dave and our clinical colleagues that we actually have a pathway to do that at Monash, which is fantastic. And it's about getting the right agent into the patients. And for me, that will be the highlight of the next phase of my career. But Pouya, you're already there, so.
He's waiting for the calls.
So to be honest, I really like to do something for patients who have DIPG. So it's kind of brain cancer for pediatric brain cancer, which there's no treatment for them. So we started a project on that, and I know it will be very, very challenging, but I really like at some point when somebody diagnosed by DIPG, we say, okay, this is one solution for you. And it's like a fantasy for me. I think after that, I can get retired.
We'll all be happy if you can do that, Pouya.
Well, I think what came through in both of your nominations was that, you know, there were amazing strides that had been achieved, but probably the best was still to come, and probably the impact, as you've described, as the impact on patient care is probably inevitably still to come. So I think that was the great promise in both of your work is really, really evident in the way that you presented and the way that it was presented to the panel. So congratulations to you both. If there aren't any other questions from the audience, I think we can bring the meeting to a close. But I'd just like to especially thank those that spent the time to nominate our career research scientists, particularly Roger and Tony, to prepare the two winning nominations. And to you, Renee and Pouya, I'll be more familiar now. I've been trying to keep presentations very formal, but thank you so much for the fantastic work that you've been doing. Obviously your engagement in the community, not just within the MPCCC domain, but across Victoria and internationally. I think that's part of the exciting bits to hear of your talks, and we look forward to hearing more installments as you progress forward. And thanks everyone else for attending. I'm sure you've all got some sort of sense of the energy in the two research programs that we've heard, and that we all look forward to hearing from you in the future and hearing how you've spent your prize money. So thank you everyone, and thank you so much, May, for doing the background work to this. It was a great assistance to everyone for it to be pulled off so smoothly. Okay, so thank you everyone, and have a nice afternoon.
Thanks, Melissa.
Thank you.
Good weekend.
Yeah.